We are talking about monoclonal antibodies. Firstly, we have to know the basic structure of antibodies, which consists of four polypeptide chains that contains two light and two heavy chains. Each light and heavy chain consists of two regions, the variable and the constant region. Also, we have to know the difference between polyclonal antibodies and monoclonal antibodies. While polyclonal antibodies are inexpensive, quick to produce, and generate large amounts of non-specific antibodies, while monoclonal antibodies are expensive, taking long time for production, and generate large amounts of specific antibodies. Monoclonal antibodies are better than polyclonal antibodies due to specificity of binding, homogeneity, and their ability to be produced in unlimited quantities. There are four types of monoclonal antibodies: murine, chimeric, humanized, and fully human antibodies. Murine antibodies are produced using rats by hybridoma technology. The rats antibodies differ from the human antibodies, so it may lead to some disadvantages like reduced stimulation of cytotoxicity, and these antibodies lead to mild allergic reactions. Hybridoma technology is done by purification and characterization of the desired antigen in adequate quantity, then immunization of mice with the purified antigen, then removal of spleen cells from mice and its fusion with myeloma cells using polyethylene glycol as fusing agent. Following fusion, the hybridomas were grown in a specific medium. The mixture is then diluted and the clones are grown. The secreted antibodies are then assayed for their ability to bind to the target antigen, and finally propagation of hybridoma is done in vivo or in vitro. Chimeric antibodies, the second type, which is produced by recombinant DNA as mouse DNA encoding the binding portion of a monoclonal antibody, was merged with human antibody producing DNA in living cells. It is produced by human constant region and mouse variable region. The third type is called humanized antibody, which is about 90% from the human. Humanized monoclonal antibodies composed of only the hypervariable regions of murine antibody. It is produced by recombinant DNA method with less immunogenicity effect. The last type of monoclonal antibodies is called fully human monoclonal antibodies, which is produced by phage technology and genetic engineering as recombinant DNA. It is better than other antibodies because it has less side effects and no immunogenicity response. There are many applications of monoclonal antibodies, like diagnostic applications, as several diagnostic procedures that use monoclonal antibodies are now available, like classification of blood groups in humans and the detection of causative organisms, and tumors antigen detection. Although there are therapeutic applications, as monoclonal antibodies and their derivatives can affect target cell function and viability via several different mechanisms, as monoclonal antibodies can be used to engage cytotoxic effector cells. Another application is the possible treatment for cancer involves monoclonal antibodies that bind only to cancer cells specific antigen and induce immunological response on the target cancer cell. Monoclonal antibodies can be modified for delivery of radioisotope or cytokine. Although there are other many applications for monoclonal antibodies, and one of them is using it to purify a substance with techniques which are called immunopurification and affinity chromatography. There are miscellaneous applications of monoclonal antibodies, like using monoclonal antibodies to detect pregnancy with antibody to beta HCG. There are many approved drugs in the market that consists of monoclonal antibodies. One of these drugs is called Repro, which contains the antibody apiximab. This drug is used to prevent coagulation in coronary and inhibit platelet aggregation. Another drug is called Humira, which consists of an antibody that is called adalimumab. Its isotype is human IgG1. This drug is used for autoimmune diseases and is effective in rheumatoid arthritis, Crohn's disease, ulcerative colitis, and ankylosing spondylitis. Another drug is called Avastin, and its antibody is called bevacizumab. It is an angiogenesis inhibitor. This drug is used to treat lung and breast cancer. This drug isotope is CDR grafted mouse and human IgG1. Mylotarg is a drug which was used to treat acute myeloid leukemia, but this drug was withdrawn from market as it showed increased patient death. The drug antibody was gemtuzumab. Herceptin is a drug which is mainly used to treat certain breast cancers, but also increases the risk of heart problems, including heart failure and left ventricular ejection fraction decline. The antibody is trastuzumab. Synagis is a drug which is produced by recombinant DNA technology. It is used in the prevention of respiratory syncytial virus infections. It is recommended for infants that are high risk because of prematurity or other medical problems such as congenital heart diseases.